In this video, we'll study about the histology of the general connective tissue. What is connective tissue? It is applied to a tissue that fills the interstices between more specialized elements. Next, we'll see about the components of the connective tissue. So, the three main components are, first you have the cells, then you have the fibers, and the third one is the intercellular ground substance. Now we'll see about the fibers of the connective tissue. So there are three main types of fibers are seen. One is the collagen fiber, next is the reticular fibers and the elastic fibers. So first we'll see about the collagen fibers. The collagen fibers are more numerous. They can be classified into various types. The reticular fibers were once described as a distinct variety of fiber, but they are now regarded as one variety of collagen fibers. So this is a diagrammatic representation. Here you can see these are the collagen fibers and this is the elastic fibers and these are reticular fibers. So next we'll see about the collagen fibers in detail. The collagen fibers are more abundant type of connective tissue fibers. With light microscope, the collagen fibers are seen in bundles. The bundles may be straight or wavy depending upon how much they are stretched. The bundles are made up of collections of individual collagen fibers which are about 1 to 12 micrometer in diameter. The bundles often branch or anastomose with adjacent bundles, but the individual bundles do not branch. With electron microscope, each collagen fiber is seen to be made up of fibrils that are about 20 to 200 nanometer in diameter. Each fibril consists of number of microfibrils that are about 3.5 nanometer in diameter. At high magnification of electron microscope, each fibril show a characteristic cross striations or periods after every 67 nanometer interval. When you see the staining characters, the bundles of collagen fibers appear white with the unaided eye. In sections stained with hematoxylin and eosin, the collagen fibers are stained light pink. With special methods, they assume different colors depending upon the dye used. Two commonly used methods are Mason's trichrome, with which the fibers stain blue, and Van Gieson, the method with which they stained red. After silver impregnation, the fibers are stained brown. When you see the chemical nature, the collagen fibers are also called because they are made up of mainly a protein that is called as collagen. The collagen is made up of molecules of tropocollagen. The microfibrils of collagens are chains of tropocollagen molecules. Next we'll see about the physical properties. The collagen fibers can resist considerably the tensile forces, that is the stretching, without significant increase in their length. At the same time, they are pliable and they can bend easily. When polarized light is thrown on the fibers, the light is split into two beams that are refracted in different directions. This is called as birefringence. It is an indication of the fact that each collagen fiber is made up of finer fibrils. The collagen fibers swell and become soft when treated with a weak acid or alkali. They are destroyed by strong acids. On boiling, the collagen is converted into gelatin. Next, we'll see about the production of the collagen fibers. The tropocollagen is synthesized by fibroblasts and released into extracellular spaces where they get polymerized to form collagen fibers. The collagen is not only synthesized by the fibroblast but by other cells also. Example, the chondroblast in cartilage, the osteoblast in bone, and smooth muscle in blood vessels. Next we'll see about the varieties of collagen and their distribution. Several types of collagen, more than 25, are recognized depending upon the diameter of the fibers, the prominence of the cross striation and other features. So the types are mainly the type 1. So these are collagen fibers of classical description having the properties described above. They are found in connective tissue, tendons, ligaments, fasciae, aponeurosis, etc. They are also present in the dermis of the skin and in the meninges, and they form the fibrous basis of the bone and of the fibrocartilage. The type 1 fibers are of large diameter, about 250 nanometer, and they have a prominent cross striation. Next is the type 2. The type 2 are of two subtypes. So they have a larger one, and this is about 100 nanometer in diameter, while the narrow fibers, they are about 20 nanometer in diameter. In type 2 collagen, the striations are less prominent than in type 1. The type 2 collagen fibers form the fibrous basis of hyaline cartilage. The fine type 2 fibers also present in the vitreous body. Next is the type 3. The type 3 forms the reticular fibers. So this we'll see later. Then the type 4. So this type of collagen fibers consist of short filaments that form sheets. It is present in the basal lamina of the basement membrane. It is also seen in the lens capsule. Then the type 5. This type of collagen is found in the blood vessels and in the fetal membranes. Next we'll see about the reticular fibers. These fibers are a variety of collagen fibers, and they are composed of collagen type 3, and they show periodicity, that is the striation of 67 nanometer, and they differ from the typical, that is the type 1 collagen fibers as follows. They are much finer and they have uneven thickness, and they form a network by branching and by anastomosing with each other. They do not run in bundles. They can be stained specifically by silver impregnation, which renders them black. Thus can be easily distinguished from type 1 collagen fibers, which are stained brown. Because of their affinity for the silver stain, reticular fibers are sometimes called as argentophil fibers. The reticular fibers contain more carbohydrates than type 1 fibers. The reticular fibers provide a supporting network in the lymphoid organs like the spleen, lymph node and bone marrow, and most glands including the liver and the kidney. The reticular fibers form an essential component of all basement membrane. They are also found in relation to smooth muscle and nerve fibers. The reticular fibers are synthesized by the fibroblast and the reticular cells. Next we'll see about the elastic fibers. The elastic fibers are fewer than that of the collagen fibers. They run single, not in bundles. They branch and they anastomose with other fibers. The elastic fibers are thinner than those of the collagen. This is about 0.1 to 0.2 micrometer in diameter. In some situation, the elastic fibers are thick. Example, in ligamentum flava. In other situations, as in the wall of the large artery, they form the fenestrated membrane. With electron microscope, each elastic fiber is seen to have a central amorphous core and an outer layer of fibrils. The outer fibrils are made up of glycoprotein that is known as the fibrillin. The periodic striations are not present in elastic fibers. When you see the staining characters, the elastic fibers do not stain with usual stains for collagen. They are demonstrated by staining with orsin and with aldehyde fusion and by Verhoef's method. When you see the chemical nature, elastic fibers are composed mainly of a protein that is called as elastin that forms their center amorphous core. The elastin is made up of smaller units that is called as the tropoelastin. Next we'll see about the production of the elastic fibers. The elastic fibers of connective tissue are produced by the fibroblast. In some situation, the elastic tissues can be formed by the smooth muscle cells. Next we'll see about the physical property. As their name implies, the elastic fibers can be stretched and return to their original length when a tension is released. They are highly refractile and are therefore seen as a shining lines in unstained preparation. The relaxed elastic fibers do not show birefringence, but when stretched, the fibers show highly birefringent. Unlike collagen, the elastic fibers are not affected by weak acids or alkalis or by boiling. However, they are digested by the enzymes, the elastase. Next, we'll see the cellular component of the connective tissue. Various types of cells are present in the connective tissue. They can be classified into two distinct categories. The cells that are intrinsic components of the connective tissue and the cells that belong to the immune system, and they are closely identical or related with certain cells present in the blood and in the lymphoid tissue. So the cells that are intrinsic component of the connective tissue are the fibroblast, undifferentiated mesenchymal cells, the pigment cells and the adipocytes. The cells that belong to the immune system are macrophage cells, the mast cells, the lymphocytes, the plasma cells, the monocytes and the eosinophils. Next we'll see about the fibroblast cells. So these are the most numerous cells of the connective tissue. They are called fibroblasts because they are concerned with the production of the collagen fibers. They also produce reticular fibers and elastic fibers. Where they are associated with reticular fibers, they are usually called as reticular cells. The fibroblasts are present in close relationship to the collagen fibers. They are fixed cells, that is, they are not mobile. In tissue section, they appear to have a spindle shape and the nucleus appears to be flattened. When seen from the surface view, they show branching processes. The nucleus is large, euchromatic and has a prominent nucleolus. The amount of cytoplasm and of the organelles varies depending upon the activity. In inactive fibroblast, that are the cytoplasm scanty, organelles are few and the nucleus may become the heterochromatic. Inactive fibroblasts are often called as the fibrocytes. In contrast to the fibrocytes, the active fibroblasts have abundant cytoplasm. The endoplasmic reticulum and the Golgi complex and the mitochondria become much more prominent. The fibroblasts become very active when there is a need in lay down collagen fibers. This occurs, for example, in wound repair. When the need arises, the fibroblast can give rise by division into more fibroblasts. Next we'll see about the myofibroblast. Under electron microscope, some cells resembling fibroblast have been shown to contain actin and myosin arranged as in smooth muscle, and they are contractile. They have been designated as myofibroblast. In tissue repair, such cells probably help in the retraction and the shrinkage of scar tissue. These cells are pluripotent and they develop into new cells when stimulated. They are found along the periphery of the blood vessels, hence they are also called as the adventitial cells. Next we'll see about the undifferentiated mesenchymal cells. Embryonic connective tissue is called mesenchyme. It is made up of stellate shaped cells with a slender branching processes that join to form a fine network. This is called as undifferentiated mesenchymal cells. Next we'll see about the pigment cells. The pigment cells are easily distinguished as they contain the brown pigment that is called as the melanin in their cytoplasm. They are most abundant in the connective tissue of the skin and in the choroid and in the iris of the eyeball. Along with the pigment containing epithelial cells, they give the skin, the iris and the choroid their dark color. Variation in the number of the pigment cells and in the amount of the pigment in them accounts for the differences in the skin color of different races. Of many cells that contain the pigment in their cytoplasm, only a few are actually capable of synthesizing melanin. Such cells are called as melanocytes and they are of neural crest origin. The remaining cells are those that have engulfed the pigment released by other cells. Typically, such cells are star-shaped cells, the stellate cells with a long branching processes. In contrast to the melanocytes, such cells are called as the chromatophores or melanophores. They are probably a modified fibroblast. The pigment cells prevent light from reaching the other cells. The importance of this function in relation to the eyeball is obvious. The pigment cells in the skin protect the deeper tissue from the effects of the light, especially the ultraviolet light. Next we'll see about the fat cells or adipocytes. Some amount of fat may be present in the cytoplasm of many cells including the fibroblast. Some cells store fat in large amount and they become distended, and these are called as fat cells, adipocytes or lipocytes. Aggregation of the fat cells constitute the adipose tissue. Each fat cell contains a large droplet of fat that almost fills it. As a result, the cells become rounded. The cytoplasm of the cell forms a thin layer deep to the plasma membrane, and the nucleus is pushed against the plasma membrane and it is flattened, resembling a signet ring. The adipocytes are incapable of division. Next we'll see about the macrophage cells. The macrophage cells of connective tissue are a part of large series of cells present in the body that have the similar function. So these are collectively known as the mononuclear phagocyte system. The macrophage cells of the connective tissue are called as histiocytes or plasmocytes. They have the ability of phagocytose unwanted material. Such material is usually organic. It includes bacteria invading the tissue and damaged tissues. The macrophages also phagocytose inorganic particles injected into the body, example dyes. In ordinary preparation of the tissue, it is difficult to distinguish macrophages from other cells. However, if an animal is injected with India ink or tryptophan blue or lithium carmine particles, these are taken up into the cytoplasm of the macrophages, thus making them easily to recognize. The macrophages are usually described as fixed cells when they are attached to the fibers, or motile or free cells. The fixed macrophages resembles fibroblast in appearance, but free macrophages are rounded. However, all macrophages are capable of becoming mobile when suitably stimulated. The nuclei of the macrophages are smaller and they stain more intensely than those of the fibroblast, and they are often kidney shaped. With the electron microscope, the cytoplasm seen to contain numerous lysosomes that help in digesting material phagocytosed by the macrophages. Sometimes the macrophages may fuse together to form a multinucleated giant cells. Apart from direct phagocytic activity, the macrophages play an important role in immunological mechanism. Next we'll see about the mast cells. The mast cells are small, round or oval cells. These are also known as the mastocytes or the histaminocytes. The nucleus is small and centrally placed, and you can see the irregular microvilli, that is known as the filopodia, are present on the cell surface. The distinguishing feature of these cells is the presence of numerous granules in the cytoplasm. The granules can be demonstrated with the PAS stain. They also stain with dyes like toluidine blue and Alcian blue. With them, the nuclei stain blue, but the granules stain purple to red. On the basis of the staining reaction, the granules are known to contain the acid mucopolysaccharides. With the electron microscope, the granules are seen to be vesicles, each of which is surrounded by a membrane. The granules contain various substances. The most important of this is histamine. The release of histamine is associated with the production of allergic reaction when a tissue is exposed to an antigen to which it is sensitive. Apart from histamine, the mast cells may contain various enzymes and factors that attracts the eosinophils or neutrophils. The mast cells differ considerably in size and in number from species to species and at different sites in the same animal. They are the most frequently seen around blood vessels and nerves. The mast cells are probably related in their origin to basophils of the blood, and they may represent modified basophil cells. Next we'll study about the lymphocyte. The lymphocyte represent one variety of leukocytes, the white blood cells present in the blood. Large aggregation of the lymphocytes are present in the lymphoid tissue, and they reach the connective tissue from these sources, and they are specially numerous when the tissue undergo inflammation. The lymphocytes play an important role in the defense of the body against the invasion by bacteria and other organisms. They have the ability to recognize substances that are foreign to the host body and to destroy these invaders by producing antibodies against them. The lymphocytes are derived from the stem cells present in the bone marrow. They are of two types, the B lymphocytes. They pass from the blood to reach other tissues directly. Some B lymphocytes mature into plasma cells, and the second type of lymphocytes are called as the T lymphocytes. They travel from the bone marrow to the thymus. After undergoing a process of maturation in this organ, they again enter the bloodstream to reach the other tissues. Both B lymphocytes and T lymphocytes can be seen in the connective tissues. Apart from the lymphocytes, two other types of leukocytes are present in the connective tissue. One is the monocytes, or closely related in function to the macrophages. Then is the eosinophils, are found in the connective tissue of many organs. They increase in number in allergic disorder. Next we'll see about the plasma cells or plasmocytes. The plasma cells is seen to be small and round, and they have a basophilic cytoplasm. It can be recognized by the fact that the chromatin in this nucleus forms 4 or 5 clumps near the periphery of the nucleus, thus giving a nucleus a resemblance of cart wheel. With the electron microscope, the basophilia is seen is due to the fact that the cytoplasm is filled with rough endoplasmic reticulum except for a small region near the nucleus where a well-developed Golgi complex is located. Both these features are indicative of the fact that the plasma cells are engaged in considerable synthetic activity. They produce antibodies that may be discharged locally, may enter the circulation, or may be stored within the cells itself in the form of inclusion that are called as the Russell bodies. Very few plasma cells can be seen in normal connective tissue. Their number increase in the presence of certain type of inflammation. It is believed that the plasma cells represents B lymphocytes that have matured and have lost their power of further division. Next we'll see about the intercellular ground substance of connective tissue. So this fills the space between the cells and the fibers. If a small quantity of fresh areolar tissue is spread out on a slide and it is examined under a microscope, the spaces between the fiber bundles appear to be empty. If such preparation is treated with silver nitrate, the spaces are seen to be filled with a brown staining material. These ground substances are rich in protein carbohydrate complex which are known as the proteoglycans. So each of them is a complex formed by a protein and a long chain polysaccharide which is called as the glycosaminoglycans. In addition to the proteoglycans, the ground substance also contain the structural glycoprotein. Their main function is to facilitate the adhesion between the various elements of connective tissue. The intercellular ground substance is synthesized by fibroblast, osteoblast, chondroblast, and even the smooth muscle cells can produce ground substance.